Okay, so we discuss nucleic acids. So these nucleic acid, the nucleic acids, they are simply coming from the word nucleus. Yeah, so this is where we get to simply talk about the nucleus. So this is where nucleic is coming from. So just a quick recap. What are the functions of the nucleus? So there are a number of them. So one of the major functions, we say the nucleus controls, it controls all cell activities. All the activities of the cells are controlled by the nucleus. So now one other thing about the nucleus is that this one, the nucleus, it is the one which controls, which stores genetic material. So when we're talking about the nucleus, we're talking about something that will contain the genetic material. Okay, so that is what will form our introduction. So we're basically dealing with something that will relate to genetic material. So now we're dealing with nucleic acids. So nucleic acids, we have two of them. So the nucleic acids that we have so this diagram, make sure you write it somewhere. Write it in your book, or write it somewhere so that you can use it up for revision. So the nucleic acids, they contain, they have DNA and RNA. Okay, so the information is flowing nicely. So DNA, which is deoxyribonucleic acids. Okay, so this A, it's acid. So remember, dealing with acids. Yeah, so I believe you know very well. So this is deoxyribonucleic acids. So one thing you will notice is this. Have you seen that this is actually our topic? Nucleic acid. So one type is this one, which starts with D, deoxyribonucleic acids. Yeah, so this is what we're basically looking at, the nucleic acid. So this nucleic acid is what we're trying to discuss. So you can take notes, write somewhere so that you follow them up. Yeah, so this is our DNA. DNA stands for deoxyribonucleic acid. And then RNA, what does RNA stand for? So RNA, it stands for ribo. So that one, it's ribonucleic acid. Okay, so we have our ribonucleic acid. Yeah, so I don't even have much space. So again, one thing you take note of is this nucleic acid. Are you seeing it's coming out everywhere? So one thing which you now take note of is Na. N simply means nucleic acid. So this is our topic, nucleic acid. You have seen DNA, or Na and Na. The only difference is this D and R. One is deoxy, and then the other one is ribo. And then now the main difference between these two are the ribonucleic acid and direct the DNA. So the main difference is like this one, the ribonucleic acid, it it's coming from, we can relate it to ribosomes. So we know ribosomes very well. So ribosomes are very tiny structures of the cell, which which uh, which play a role in protein synthesis. So the ribosomes, they work very well. Their main specialty, they are specialized in dealing with proteins. So here where we have ribonucleic acid, the RNA plays a role in protein synthesis. And in the DNA, this one is very common. We have heard a lot about it. The DNA, it stores genetic information, so it mainly deals with heredity. Okay, so this is our foundation, and then from here now we advance. Yeah, so let us advance. Okay, and what do I mean by advancing? Yeah, we're now going further. So now we can talk about these two. So like, what are the key differences between the DNA, and just what are the things found on the DNA and also on the RNA? So some of the things we can take note of. So I emphasize, kindly write down these. Uh, yeah, so write down what we're discussing. So the DNA. Yeah, I'll start with the DNA. So one thing about the DNA is that it forms a double helix. So this is double. So the DNA forms a double helix. So like we usually have two strands of DNA. So this is part 1 of our lesson. So part 2 later on, we'll be able to discuss translation, yeah, all those processes of DNA transcription, yeah, all those. So DNA, it is a double helix. Yeah, so it forms two strands, and like RNA, we're going to look at it, it's just one strand. Yeah, so this is one thing. So the DNA is double, it has double strands, it's a double helix structure. But for the RNA, it is like most of you, it is this. So at some people be offended. Hmm. Yeah, it's okay to be RNA. So I think for just a short break. So I'm sure you, by playing this voice note, you are familiar with the Megaminds documents. Yeah, so if you don't have them, you can look out for them. So this singleness, it also related something about elements. So I just wanted to remind you about that. So now here, RNA has single strands. Okay, and then DNA is double strands. And then we can talk about what can we say? Okay, so we can go to, so I'm just trying to compare and contrast. So like in DNA, this is where there is replication. So in DNA, that's where genes are replicated, if I would say that. Yeah, so DNA, there is replication. There is DNA replication, and then there is also storage of genetic information. So genetic information is stored. Yeah, so storage. So under DNA, there is storage of genetic information. This is falling under DNA. And then on the other side, where we have RNA, on the RNA, we find transcription. So RNA, that's where we have transcription. Yeah, so the handwriting might be bad, but I guess the voice is clear. Okay, so uh, this is transcription. Yeah, so pardon my bad handwriting. I'll get the excuse that I'm a medical student, so under my handwriting is this bad. Okay, and then we have transcription and also translation. So if you are to contrast DNA and RNA, this is what you can write about. Yeah, so this is translation. Okay, and then from there now, so these are things for you to make use of. Yeah, you study them, so you can take a screenshot or yeah, write them down somewhere. You should generate notes from this lesson we are having. Okay, so these are some of the key differences we can take note of. Yeah, so these are related or mainly involved to DNA, and then these guys here, they are falling under RNA. Okay, and then now from there we can now go to we complete our diagram. uh, okay, so we're now going to talk about components. Yeah, so where do we pick it up from? Okay, so let me just clear up my board. Okay, so now we're talking about nucleic acids. So just a reminder of where we are. This is nucleic acids. We have DNA and RNA. And then from here now, so the DNA and RNA, so I've listed those differences. Yeah, and then now they obviously have something in common. Yeah, so in DNA and RNA, so uh, under both of these now, we have we have nucleotides. So under DNA and RNA, we have nucleotides. So nucleotides are simply components of DNA and RNA. So the nucleotides are found, they are what make up DNA, and they also make up RNA. And then now, these nucleotides, they also have the nucleotides, they have their own components. So what we can say here, take note that nucleotides, they are monomers. They are monomers. So this is ER. So the nucleotides are monomers of DNA and RNA, meaning that they make up, when we combine different nucleotides, would either form DNA or RNA. And then these same nucleotides, they have their own components. So we're now going to explore the components of the nucleotides. So remember, remember we're starting from what the nucleic acids, of the D nucleic acids and the R nucleic acids. Yeah, so you now know that Na is nucleic acid. Yeah, this Na is nucleic acid. So ribonucleic acid, deoxyribonucleic acid. Okay, so we're now going to the components of nucleotides. So what do we find in a nucleotide? So the nucleotide has three components. So we have, in a nucleotide, we have a nitrogenous base. So just for short, I'll write it as nitrogen base. So you can finish it up and say nitrogenous. So we have a nitrogen base, and then under nucleotides we also have a pentose sugar. So we have a pentose sugar, and then we also have the last group, which is the phosphate. We have the phosphate group. So this is phosphate here. So we have our phosphate group. So these are the three things which are found in a in a nucleotide. So a nucleotide will have a nitrogen base, and then it will have a pentose sugar, and it's also going to have a phosphate group. So from here now we go deep again. So under the nitrogen base, so under this one, so under the nitrogen base, we have two components. So under the nitrogen base, we have the purines and the pyrimidines. The word will be very long and I feel so lazy. Pyrimidines. Okay, so this is what we have under the nitrogen base: purines and pyrimidines. And then for the pentose sugar, also the pentose sugar, so my body is very, very, very small. Okay, so what we're going to do is, okay, so we'll see how we can utilize it. So under the pentose sugar, so I hope you are doing all this as well in your on your sheet of plain paper, or if it's in a book, yeah, so that you can follow the way these things are flowing. Okay, so I think I won't write them in this way. I'll just list them. So under the nitrogenous base, we have purines, and pyrimidines. And then under the pentose sugar, so this is how we're flowing. Yeah, so you can actually even put them in, you can enclose them like this. Yeah, so decorate your work as neat as you can. Yeah, so don't write as bad as this. Yeah, so you can do the same for all these. You put them in the toma boxes and the like. Okay, so under pentose sugar now, uh, and just a reminder, we talked about these. Like if we have a pentose, it means pentose relates to 5. Yeah, so a pentose sugar will have 5 carbon atoms. If it's a hexose, it will be 6. Okay, so now the pentose sugar, we have 2 types. We have the ribose, sugar. So this is our ribose. Okay, I think I should at least write it better. Ribose. Okay, and then we also have the de deoxyribose. So we have I there. Okay, so we're also going to the other side. So I want to remind you on what we have discussed already. We have DNA and we have RNA. So what does Na stand for? By now you should be able to know it's nucleic acid. So here, and then this R is ribose, D is deoxyribose. Yeah, so I think DNA is like the big boss. Are you able to see that in deoxyribose we also have this ribose? So if we are dealing with math, we could have said this one is a subset of that other guy. This one is a superset. Okay, so that's just for fun. So obviously this is relating to RNA, and then this is relating to DNA. So if a question comes, you are asked that what sugar is found in DNA, you should be able to say it's deoxyribose. What sugar is in RNA? It's a ribose sugar. Yeah, we can make progress. So now, what can we talk about the phosphate group? Okay, so under the phosphate group, this one is this guy is not well known. Yeah, so our focus is mainly on these two. So key things to take note of is that the phosphate sugar, the phosphate group is acidic. Yeah, and then the phosphate group should look like this. And then if we are to draw it, we're going to have oxygen. Yeah, it is phosphorus is bonded to four oxygen atoms, yeah, and it's not even clear this side. Yeah, so we won't discuss much on the phosphate group. Yeah, so it will come in later. So we're still going. So are you seeing where we're coming from? Yeah, so that is our work. We have DNA, RNA. Both of these, they are made up of nucleotides. Nucleotides, we have nitrogenous base, pentose sugar, phosphate group. Under this, we have that. Okay, so I hope we are together up to this point. What's next now? What's next is to dig deeper again. So we keep digging deeper. Yeah, so we have listed purines and pyrimidines. So again, under purines and pyrimidines, we have some components. Okay, so I'm now regretting writing them in this manner. Yeah, so anyway, we'll sort it out. So here under the purines, so like this, so let me just rewrite. So we have the purines, the purines there. We have the purines and pyrimidines. So you should be able to consolidate a complete flowchart. Like it should be able to show you how the information is nicely flowing. Yeah, so this is our pyrimidine. So under purines and pyrimidines, there are some things we should take note of. Yeah, so under purines, so purines are they are double-ringed nitrogenous bases. So remember where we're coming from. Purines and pyrimidines are nitrogen bases. Yeah, so that's where they come from. So the purines, they have double rings, while the pyrimidines, they have single rings. Yeah, at least majority. So what do we find under the purines? Under the purines, we find the adenine. So we have adenine, which will abbreviate as A, capital letter A. So we have adenine, we have guanine. We have guanine. Adenine, guanine, and then under pyrimidine, we have thymine. We have thymine. We have cytosine. And we also have uracil. So how should we put it? Okay, so uracil, will write it beside thymine. So here is why we write uracil. So we have, it's not, the slash doesn't mean that the other name for uracil is thymine. Yeah, so let me even write it separate. to avoid confusion. So this is uracil. Okay, so we can discuss these. Yeah, so we can talk about these these components of the nitrogenous bases. So the abbreviations, it will just be similar to the other. This will be T, this is C, this is U, all capital letters. So here the major thing to take note of is how these are found in nucleic acids. So the adenine and guanine, so like purines, are found in both DNA and RNA. And then for the pyrimidines, not all of them are found everywhere. So for the pyrimidines, we we have thymine is found in DNA, and then uracil is found in RNA. So the rest of the things that are found in both. So like adenine is found in both DNA and RNA, guanine is found in both, and cytosine is found in both. Okay, so these are what we call the nitrogenous bases. Yeah, so like they are under nitrogen base. Okay, and you have now taken note of where they are found. If we have a DNA strand, then there will be thymine. So later on, we'll talk about transcription, whereby if we have something like, let's say we have, if we have adenine, and then we have uh, we have guanine, and then we have thymine and cytosine. So all of these are, so like this is a DNA, this should be a DNA molecule because we have thymine. So later on, when we'll be dealing with translations, transcriptions, yeah, so like these, there'll be change. So like what is thymine will be, it will be replaced with U. So in DNA, what is thymine, there'll be uracil when we're talking about RNA. Okay. So where are we? Oh, so we have listed down the things which are found in RNA and DNA. So these are the nitrogenous bases, and yeah, you just have to know that they are found all over. And then there are also what we call base pairing. So base pairing is where we have to know like DNA. Yeah, so like DNA, you know that it's a double helix. Yeah, so like DNA is a double helix. So like there is pairing. So like if we have thymine, and then if we have another DNA strand, this side we would have, if this is adenine, it will be paired to thymine. Okay, so that's what we want to talk about, the base pairing of these nitrogenous bases. We can start from, yeah, we'll start with the same adenine and thymine. Okay, so we can discuss that it's basically the complementary base pairs. So like we have to know what pairs, which one complements what. So like we're going to use a mnemonic, yeah, for us to get the concept very well, and not to forget. Yeah, so our mnemonic will be, what are we going to put as our mnemonic? So our mnemonic will be O O all the girls, what am I writing? All the girls cried. All the girls cried. So this will be our mnemonic. Uh, okay, so all the girls cried. Uh, is it? Oh yeah, it will make sense. So all the girls cried. This will be our mnemonic to understand the base pairs. Yeah, the complementary bases. So you can take note that uh, we have all the girls cried. Yeah, so this is this is the base complementary for DNA. Yeah, so all the girls cried. A is adenine, this is thymine, guanine, and cytosine. So adenine pairs with thymine, and then guanine pairs with cytosine. So can you surely forget? Yeah, so I'm positive you wouldn't. So all the girls cried. Adenine, thymine, guanine, cytosine. Yeah, and you can come up with one of your own whereby you say all the guys cheated. Yeah, you'll still end up with this. All the guys cheated. And then you'll be able to remind yourself, oh, so adenine with thymine, guanine with uracil. Oh, sorry, guanine with cytosine. So when you just know one, then you can't forget the other one. Because remember what we said, so this is DNA. This is in DNA. So if we are to talk about RNA, it will just be the same, except where there is thymine here, we're going to put uracil, and then this will just be like that. So when we have RNA, adenine pairs with uracil, and then guanine still with cytosine. So I think guanine is this one. Guanine is a good guy, unlike ACK. No, this one doesn't listen. When we have DNA, adenine will be busy with thymine. When we have RNA, this adenine should go to uracil. So these are bad manners. I think I don't like adenine anymore. Okay, but we're together, aren't we? So these are the base pairs. From here we can, what can we talk about? So let me just scroll up. Let's see, what have we discussed. Also, we're here on the purines and pyrimidines, and we did this. So from here we can talk about this, the pentose sugar, and then that will be the end of part 1. So just for revision's sake, this is in DNA. So this is DNA. This one here down will be RNA. Okay, so now we can quickly talk about the sugars. Pentose sugar, ribose sugar. Yeah, so this is our anagram, if I would call it like that. So let's discuss the pentose sugar, and we can call it a night for today's lesson. So on the pentose sugar, one thing you take note of is that it will have 5 carbon atoms. Yeah, so a pentose sugar can look like this, as if it's a star shape. So this is oxygen, and then we have carbon 1, we have carbon 2, carbon 3, carbon 4, and then carbon 5 will be there. Yeah, so that is the basic structure of the pentose sugar. And then the difference is on carbon number 2. So like we now want to differentiate this pentose. We have a pentose ribose sugar. So this is a pentose. So this pentose sugar can either be a ribose, or it can be a deoxyribose, blah 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 blah. Okay, so we want to differentiate. How do you know that this is a ribose, or how do you draw it? So the difference is on carbon number 2. So for the ribose sugar, on carbon number 2, we have hydrogen on top, and then oxygen it's found down there. This is our ribose sugar. Yeah, and then you should be able to remember the basic structure of glucose. Yeah, those sugars which we did. Yeah, so I hope you went through the Megaminds document and you're able to do all those. Yeah, so my interest wasn't here. Yeah, doing all these. Yeah, so I just wanted to highlight to you. So like I'm guessing you know how to draw the pentose sugar. Yeah, okay, but let me not be that lazy. So our pentose sugar can look like this. Here we have hydrogen, hydroxide, and then also as you are preparing for your exams, take note of everything we've at least tried to discuss. So remember, what does it mean when we have an alpha glucose? What is a beta glucose? What is L glucose, D glucose? Yeah, so all those you should be able to remember. Yeah, so this is why you have to study. Yeah, so I believe you are doing your best. So this is our ribose sugar. Here we're going to have H, and then this side you have something like this. That was so tiring. That was tiresome. So the idea was here we have a hydroxyl group. This is a ribose sugar. Yeah, so this is a ribose sugar. And then for a deoxyribose, it will look very similar. So everything just very similar, but the difference will be here on carbon number 2, instead of having a hydroxyl, would have just hydrogen. So when we put hydrogen, it means that's a deoxyribose. Yeah, so no hydrogen. So that's the structural difference between a ribose sugar and a deoxyribose. So here we have H. Wow, this is so familiar. Uh, this guy. This guy. Yeah, so we're done with our lesson for tonight. Yeah, so I would like to conclude with an encouragement after seeing this. The seventh president of Zambia, Hakainde Hichilema. So let me share with you a brief background. Like, do you know his story? So for those who would want to give up on whatever they are doing, school is pressurizing you, you are going through all that. No, yeah. So pressures in life will always be there. It can be a side hustle, your business, something like that. Things are not going well, and your options are just like, no, I'll give up. So be like So do you know this guy he became the UPND president when most of you watching this video when you are not born. I think that was somewhere in 2001. That's when he became the leader of this party, UPND. So what I want to share with you is his journey, like how it was. It was a very rough journey. He started contesting in when was that? I think I was how old? Yeah, I was very big. So he started contesting in 2006. He wanted to become the president of Zambia, but what happened in 2006? He lost. Imagine. You can cry. 2006, he lost the elections. He lost to Mr. Levi Mwanawasa. And then, just like your map says, try again, try again. 2008, Mwanawasa passed on, and contested. I'm sure by that time was like, no, but this is my year. What happened in 2008? Ngwang. 2008, Mr. lost. But this guy was a fighter. After these by-elections, again he came 2011. He was like, well, this is my year. I shall win. But what happened? We had Benamai Kosata. HH lost. So how many times are we counting? And you know where the story is going. Imagine all these losers. If it were you, would you continue? Honestly, look at the years passing by. Some of you, that's when you were born. He was just losing. But he was a very good leader of the opposition. So he never gave up. And what did he do? I think the next time was in 2014 when this guy who beat him here died. When Mr. Sata died, HH tried again, and by this time, I honestly thought he would win because we didn't know Mr. This one. Majority of us didn't know him very well. He wasn't popular. But for the legacy that the one who won here, that legacy, it went on and people voted. And Mr. HH did the usual. Okay, how can you feel, eh? He lost. Wait, is this correct? He lost in 2006, 20 that. Oh, I think I was making a I've just made a mistake. Okay, so let's see. 2006 he lost. Yeah, 2008 he lost again. 2011 he lost. Also, this time in 2014, that's when Sata died. HH lost again in 2015 when the elections were held. Yeah, even if we change the year, he still lost. Yeah, so this life is so unfair. So this is trial number what? 4. What happened? We went to 2016. These were one of the tightest. They are the 2016 general elections, the highly contested one, whereby you have to win by 50% plus 1, unlike in these previous years. So at this point, HH thought he had it, like, this is my time. There's no way I'll lose again. And the person who beat him in 2015, that he was contesting for the first time, but HH had been losing. So obviously he thought he would win, but he lost again. 5-time loser. Yeah, so such can be life. Yeah, and he never accepted these results. Yeah, 2016 was full of drama. That's when he was even arrested in 2017. Yeah, a lot happened. There were treason. Yeah, because he was shaken, like, I've lost too much. There's no way this is true. But what happened? He had to accept defeat, and again he was trying for the 6th time. So for the 6th time, so by 2021, by then, some of us were even done with school. 2021, what happened? Okay, so this was Mr. HH breakthrough point. Yeah, so this was his year and he won by a very margin. I think that was 59% of the vote. And usually, like when he was being beaten by Mr. Lungu was winning by 50 in 2016. In 2015, he just won by 49%. But when HH won, it was big. So this should tell you something. When your time has come, Yeah, nothing will hinder you. So we all have different races to run. Don't compare your number one, or don't compare your step one to someone else's step five. Yeah, so you all run different races. Yeah, so be encouraged by the story of Mr. HH. Yeah, so your first try doesn't define you. Just because you lost when you tried at first, it doesn't mean you keep on losing. Yeah, times will be hard. You can have such a bad record, but doesn't mean that you keep on losing. Yeah, some of you, you must have written your test one. It was looking like this. Quiz one, it was looking like this. What? It was like that. So don't be demotivated and don't be so scared. Yeah, you have everything you have what it takes to make it. Yeah, so just keep on studying. Uh, your time is coming. You don't know what's ahead. So prepare for the upcoming tests. Prepare for the upcoming quizzes. The exam is waiting for you. Yeah, everything is possible. Yeah, so just keep on working hard. All is possible. Yeah, so just like HH had his year of miracle. Yeah, yeah, he had his year of breakthrough. May this year be your year of breakthrough in whatever you are doing. All the best in whatever. Have a good night. Oh, and I think we'll end here. So later on you should finish nucleic acids part 2. I hope I didn't overbore you with my story at the end. Yeah, so let's continue next time.